Good day, this is Professor Will, CBMD PhD, and today is Monday, April 4th, 2022. It is 5:46 and 22 seconds, a.m. Eastern Standard Time. Today's research, clogged arteries, so let's hop to it here. Researchers find new strategy for preventing clogged arteries. Date April 1st, 2022. Source, Albert Einstein College of Medicine. Revving up a process that slows down as we age may protect against atherosclerosis, a major cause of heart attacks and strokes. The scientists have successfully minimized artery narrowing plaque in mice that would otherwise develop those lesions. The researchers did so by boosting chaperone-mediated autophagy, a cellular housekeeping process. Research has shown that they need cellular-mediated autophagy to protect against atherosclerosis, which becomes severe and progresses when cellular-mediated autophagy declines, something that also happens when people get older. But equally important, research has proven that increasing cellular-mediated autophagy activity can be an effective strategy for curbing atherosclerosis and halting its progression. The cellular-mediated autophagy keeps cells functioning normally by selectively degrading the many proteins that cells contain. In cellular-mediated autophagy, specialized chaperone proteins bind to proteins in the cytoplasm and guide them to enzyme-filled cellular structures called lysosomes to be digested and recycled. Research has deciphered many of the molecular players involved in cellular-mediated autophagy and shown that cellular-mediated autophagy, through its timely degradation of key proteins, regulates numerous intercellular processes, including glucose and lipid metabolism, circadian rhythms, and DNA repair. Researchers also found that disrupted cellular-mediated autophagy allows damaged proteins to accumulate to toxic levels, contributing to aging. And when the toxic buildup occurs in nerve cells to neurodegenerative diseases, including Parkinson's, Alzheimer's, and Huntington's disease. And cardiovascular disease is the world's leading cause of death, with more than 80% of those cardiovascular disease deaths due to heart attacks and strokes. The cardiovascular disease, in turn, is usually associated with atherosclerosis, the buildup of plaque, a sticky material consisting of fat, cholesterol, calcium, and other substances within the walls of arteries. Accumulating plaque hardens and narrows arteries, preventing them from delivering oxygenated blood to heart muscle, leading to heart attacks, the brain, strokes, and to the rest of the body. Researchers promoted atherosclerosis in mice by feeding them a fatty Western diet for 12 weeks and monitoring cellular-mediated autophagy activity in plaque-affected aortas of animals. The cellular-mediated autophagy activity initially increased in response to the dietary challenge after 12 weeks. However, plaque buildup was significant and virtually no cellular-mediated autophagy activity could be detected in the two types of cells. Macrophages and arterial smooth muscle cells that are known to malfunction and atherosclerosis, leading to the buildup of plaque within arteries. And cellular-mediated autophagy seem to be very important in protecting macrophages and smooth muscle cells, helping them function normally despite the pro-atherosclerotic diet, at least for a while, until their cellular-mediated autophagy activity basically came to a halt. Researchers noted that feeding a high-fat diet to mice totally lacking in cellular-mediated autophagy activity produced even stronger evidence of cellular-mediated autophagy importance. And plaques nearly 40% larger than those in control animals that were also on the high-fat diet. And some patients who have had strokes undergo surgical procedure known as carotid endarterectomy that remove plaque affected segments of the carotid arteries to reduce the risk of second stroke. The researchers analyzed cellular-mediated autophagy activity in carotid artery segments from 62 first-stroke patients who were followed for 3 years after their surgery. Those patients with higher levels of cellular-mediated autophagy following their first strokes never get a second one, while second strokes occurred in nearly all the patients with low cellular-mediated autophagy activity. And this suggests that your cellular-mediated autophagy activity level post-endarterectomy can help in predicting your risk for a second stroke and in guiding treatment, especially for people with low cellular-mediated autophagy. The study is the first to show that turning up cellular-mediated autophagy can be an effective way to prevent atherosclerosis from becoming severe or progressive. In summary, researchers developed drug compounds that have shown promise for safety and effectively increasing cellular-mediated autophagy activity in most mouse tissues and in human-derived cells. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.